Amniocentesis. The surgical insertion of a hollow needle through the abdominal wall and into the uterus of a pregnant female and the aspiration of fluid from the amniotic sac for analysis. Examination of the amniotic fluid itself as well as the fetal cells found in the fluid can reveal such things as fetal sex, the significant factor in inherited diseases that are sex-linked, chromosomal abnormality, and other types of potential problems. The procedure, generally carried out in the 15th to 17th week of gestation, is relatively painless and can be carried out under local anesthesia. Once used only in medical management of erythroblastosis fetalis, a blood disorder of the fetus and newborn that is caused by antibodies in the mother's blood. Amniocentesis was first performed in the 1930s. More than 50 metabolic diseases can now be diagnosed by use of the procedure, but it is used most often for the identification of chromosomal anomalies and neural tube defects. It is also often recommended for women 35 years or older and for women who have experienced 3 or more spontaneous abortions. Blood count. Laboratory test that determines the number of red blood cells, erythrocytes, and white blood cells, leukocytes, in a given volume of blood. The readings vary with sex, age, physiological state, and general health. But the blood of a normal individual contains on average 5 million red cells and 7,000 white cells per cubic millimeter. A differential blood count is the percentage of each type of white blood cell per 100 white cells counted. The white cells of a normal adult are about 55% neutrophils, 30% lymphocytes, and small percentages of eosinophils, basophils, and monocytes. A decrease in the number of red blood cells is usually associated with anemia, and an increase or decrease in the number of white blood cells can occur with infections, inflammatory conditions, or leukemia. A blood count may also include a determination of the number of platelets, the volume by percent of red blood cells in whole blood, known as a hematocrit, the sedimentation rate of the red blood cells, the hemoglobin concentration of the red cells, and the blood typing. Classification of blood in terms of distinctive inherited characteristics that are associated with the antigens located on the surface of red blood cells, erythrocytes. The ABO and the Rh blood groups are among those most commonly considered. Identification of these determinants has become indispensable in connection with blood transfusion, because the recipient and donor must have the same or compatible blood groups. Otherwise, hemolysis, destruction, or coagulation, clotting, results from interaction of an antigen on the red blood cells of one with an antibody in the serum of the other. In addition, blood typing serves to promptly identify the cause of such disorders as erythroblastosis fetalis, hemolytic disease of the newborn, which results from blood group incompatibility between mother and fetus. Since blood group determinants are inherited according to generally known mechanisms of heredity, blood typing sometimes provides a method for resolving cases of disputed paternity.